Hello everyone. Welcome to the second talk of our symposium, Eco-evolutionary Dynamics and the Making of Biodiversity. In this talk, we have the honor to have Dr. Angélica Cibrián-Jaramillo as a speaker. Angélica is an associate professor at Langevio Simvestab, that is Laboratorio Nacional de Genómica para la Biodiversidad, which is located in Mexico. There she leads a research group studying genomics and evolutionary ecology of plants. The lecture will last about 20 minutes, and then we'll have about 5 more minutes for questions, which are very welcome. So if you have any question for Angélica, you may post it on the chat, on the Zoom chat. You can do it both during the talk or right after it. And also you can do it both in English or Spanish. Okay, Angélica, I think we are ready for your talk now.
Thank you so much. Thanks for the invite. I'm going to share my screen now. Okay. All right. Okay, well, I will talk to you about a system that I find fascinating, both from an ecological perspective and an evolutionary perspective. And it has relevance for this session on eco-evolutionary dynamics in the making of biodiversity. Perhaps not so clear at first, but I hope that at the end of the talk it'll become clear. So there we go. This is the result of collaborative work with Paco Barón at Langevio as well, and a lot of students and a few postdocs that have worked very hard for the last 3 or 4 years on this system, and maybe even a little bit longer. So the main message that I want you to take from this, and I will come back to this later toward the end of the talk, is that multi-trophic eukaryote interactions are mediated by microbes and are themselves in the system an eco-evolutionary dynamic that over time maintains diversity. And I will focus specifically on cycads and their symbionts as an example of what I mean by this. So cycads are gymnosperms. They're from the order of Cycadales. They're subtropical. There are not many species. There are 250 species or so described, but they're fascinating for many biological reasons that I will go into detail in a little bit. They can live up to 2,000 years as individuals and even maybe longer to 5,000 years in some Dayun Mexican cycads. The lineage can be best described in the following way. This lineage endured a pair of cataclysms where most life was killed off the planet. This is a line from the New York Times from this year, and that's essentially what cycads are. They are living fossils. They have survived several mass extinctions. Currently, unfortunately, they're the most threatened groups of plants. They're not surviving humans very well, but they can still, when they're found, they can still thrive in soils with low nutrients or poor nutrient availability, where other plants can simply not survive. So cycads have retained many of the ancient features that characterize fossils that have been found even recently of 280 million years old. They have what we call the monoxylic pachycaul stem, which is tolerant or even resistant to fire. They have girdling leaf traces, which are an unusual trait for plants. Circinate vernation, that means that the leaves open very much like ferns. This is another ancient trait that they have maintained. They're dioecious, and they have exposed seeds in some of the genera like Cycads. And they are characterized by these fascinating and huge, often huge cones, female or male cones. But perhaps most fascinating is their associations with insects. They have more than 100 million year old associations with pollinators that are root site dependent. That means that they reproduce within the plant. And they have maintained these mutualisms for many years. They have developed an ancient beta-myrcene pull-push pollination, which is another very ancient trait in plants, way before angiosperms developed various mechanisms to attract pollinators. They also have associations with lycinid butterflies that are, again, more than 100 million year old records of these associations. And these are aposematic herbivores that are chewers, and they eat the leaves, and presumably they retain cycad toxins in their wings to protect from from predators in general. I'm going to go into details a little bit later than this. Another fascinating feature about cycads is that they have quite the particular chemistry. They are unique among many other plants in that they're highly, highly toxic, full of glycosides, cytosines, glycosides, and other non-proteinic amino acids such as BMAA. And because of this, cycads have captured both the imagination of the fiction and nonfiction writers, but also the medical community, because they have been associated to a neurodegenerative disease in humans, ALS, PDC, in some of the populations in the Pacific, in particular in Guam, where the consumption of cycads was associated with these diseases. And this is the reason, because this is suspected, is because BMAA, which is present throughout the plant, is an agonist of the glutamate receptors in the mammal brain, and therefore causes all sorts of trouble when it bioaccumulates in the body. However, our research has shown that this cycad intoxication hypothesis is something that we still have to revise and revisit, because we have generated ethnographic evidence of historical and current use of cycads as food sources, and with a very high cultural significance for at least 5,000 years in Mexico. And there are records of their use throughout the world, not only in Mexico, and with processes that detoxify cycads. But that said, cycads are still very toxic. So one of the most toxic organs that cycads have is the coralloid root. And this root is fascinating to us for several reasons. It's a specialized apogeotropic root. That means that it grows above ground, or it's capable of growing above ground. It's able to fix nitrogen, and it hosts cyanobacteria. And this is something that has been studied since the 19th century. But only recently, from a few years ago, our group and a Chinese group began to really go in depth into this organ to look and try to describe not just the cyanobacteria that are present, but other bacteria that we have found there. So we have been finding out, and this is something that I will talk to you in the next few slides, that this root not only fixes nitrogen, but it also produces a diverse source of metabolites, both by the cyanobacteria, other bacteria, fungi, and viruses, although today I'm only going to talk about bacteria. And this is the organ of the plant that has the most BMAA in it. This is a cross-section of a coralloid root by our lab. You can see here what's called the cyanobacterial zone, and this is where cyanobacteria are hosted within the coralloid root. This zone has been known for, like I said, since the 19th century, but only recently we have been describing it in more detail. So the first hypothesis that I want to describe today is that cycads can survive in nutrient-poor habitats, as I mentioned before, and also maintain these specialized biological associations by harboring endo-symbionts in the root, in the coralloid root, and acquiring their toxic metabolites, toxic for us mammals, but not necessarily for bacteria, and not necessarily for the plant. And they do this through this specialized organ. And maybe this is the key, part of the key to how cycads have been able to adapt in a very plastic manner throughout time, despite their long-lived, slow growth and slow lives in general as eukaryotes. Through their bacteria and the root, they're able to adapt quickly to changes in the environment. And the second hypothesis that we have put forward relatively recent, and this is inspired by Esperanza Martinez and her work with endophytes transfer from mammals to plants to the soil, is that these key endosymbionts that we find in the root are transferred and maintained in the entire cycad system. And when I say cycad system, I mean the insects, both the herbivores, the pollinators, perhaps other animals that we haven't described yet, the soil and all the microorganisms in the soil, and the plant itself. And so this is the second hypothesis that we have put forward. So we have done extensive fieldwork to find coralloid roots and cycadivorous insects, mostly in Mexico. Mexico is one of the biodiverse sites for cycads. And so we have there 3 genera with enough species for us to do comparative biology. And we have done a combination of fieldwork, greenhouse, microscopy, wet lab, and big data, and have basically thrown all the possible techniques to describe the system. So I'm going to tell you a little bit about what we have so far on this system. Some of it is published, but most of the things that I will mention today are not. So perhaps among the many strategies that we have developed to study this system, which is not easy because you have to go and find them, they're difficult, and you have to dig them out of the soil, often below rocks, and then you have to keep everything alive so you can get to the lab in time. So this posed a challenge, and so one of the things that we have done to study these organs, not just the coralloid root, but then eventually also the insects, is that we opted for what we call the eco-mining strategy. And this is essentially a combined strategy of, from an environmental sample, doing 16 RNA, ribosomal RNA, Illumina I-tags, as traditionally done, also axenic isolation, again, as traditionally done. But then we have this new or relatively new method that we called subcommunity co-cultures, in which we enrich for one of the groups and then allow the rest of the bacteria that are associated to that group to grow without either isolating it or just describing it without the live organisms, as we do with the amplicons. So this has allowed us to explore many questions that we cannot answer with either strain isolation, axenic strain isolation, or with other strategies of either metagenomic or 16S or environmental sampling. And from there, once we get information about the bacteria that are alive in these subcommunity co-cultures, we can actually ask questions about their function because these bacteria are actively interacting, and we can play with various conditions where we can explore different types of roles that they could be playing within the bacterial community and eventually with the plant. And from there, we've moved on to actually identifying the key players and then doing semi-synthetic communities, for example, cyanobacteria and caulobacter. And once we sequence all of this, we can go back and redo the co-culture just to tweak things that, depending on the questions that we want to ask. So briefly, what we found with this strategy is that there is a core of genera associated to the cycad coralloid root. I'm showing here results for Dioun, but we have other genera. I will mention them just briefly in a moment. And these constitute mostly cyanobacteria, as we already knew, but also other groups like Rhizobiales and a lot of other proteobacteria, including Caulobacter, which is a fascinating bacteria that I'll mention again in a moment. And there's also hundreds of other opportunists that I'm not showing here that are often there with the core on and off, depending on when and where you sample. But this is essentially the core of the coralloid root, and these are the relatively abundances of various samples here. I'm just showing you a subset of what we have so far, where the color corresponds to the various bacterial groups. Recent metagenomic assembled genomes show that the core that we find in Dioun and several species of Dioun is also found in Xenia, which is another genus in Mexico, and not only in Xenia, but also not only in the coralloid root, but also in the primary root and in the soil. And this is just an example of a few taxa that we have sequenced almost the entire genome very well, and that we're currently annotating. So this points to, again, confirming a core microbiome that is maintained both in the coralloid root, but also in the rest of the plant and the soil. But a key question here is not just who's there, but also what they are doing. So we are looking at the function of these genomes within the coralloid root. So we focus on biosynthetic gene clusters, which are linked genes that participate in a specialized metabolic pathway. And what we do is we sequence genomes and metagenomes, and then we assemble these biosynthetic gene clusters, and we look to see how they are changing either by expansion, recruitment of new genes, or deletion of genes, and how these new combinations would give rise to specialized metabolites, either of a similar in nature or actually sometimes very different, but unique to the Cycad system. So this is effectively an adaptive eco-evo dynamic response, and I will show you in a moment why I also mean that. So the key question here is, do we find biosynthetic gene clusters that are only found in the Cycad coralloid root system and that are not found anywhere else, that would suggest that they're specific of Cycads and that are doing something perhaps as a result of coevolution with a host? And so we looked at Dioun, and what you see here is a phylogeny of cyanobacteria, in which we have in green the bacteria that we assemble their genomes from coralloid roots. And this is, there's too much detail here for me to go on in it right now, but this is published work. You can read about it in the GBE paper 2019. But I just want to point out that these red squares mean that these particular BGCs are unique to the Dioun system. And some of them are found in some populations of Dioun and some are found in others. And these are other plants that host symbionts. So we find similar patterns in other bacteria, and one that's interesting is Caulobacter. This is a very small bacteria that we believe it's attached to the cyanobacteria, physically attached to the cyanobacteria. And we find that also in this species, which hadn't been reported for cyanobacteria before, of it being associated with cyanobacteria before in Cycads, has terpenes and an indigoinine-like metabolite, which has a blue color specific to the Cycad system. And this was interesting because this allowed us to actually visualize the consortia that we've been describing so far just with metagenomes and bioinformatically, and we were able to actually see it live in the microscope. So we use this indigoinine-like metabolite as an indicator to see if it turned on or off, depending on if it was with or without the cyanobacteria. And so what you have here in the first column is Caulobacter alone at different frequencies, cyanobacteria alone. And when you put them together, actually these this indigoinine-like blue color turns on. It's actually very hard to see, but it's in this very first panel. And you can see the corresponding peaks here, because basically this is just tough to see, but you can see the corresponding peaks here. It basically turns on only when cyanobacteria and Caulobacter are together, either without or without nitrogen in the media. These are part of the co-cultures that I mentioned before. This is a structure that we predicted from the BGC that corresponds to this indigoinine-like metabolite. This is an actual photo of what it looks like with the cyanobacteria with its Caulobacter turned on with this metabolite. We still don't know exactly what it's doing, but we do know it's a result of the consortia, and it's only found in this system. So another question that's key is, are these BGCs actually adapted out in the environment? Because I told you at the beginning that these might actually play a role in cycads adapting to complicated conditions that I've mentioned before. So what we did is we compared the microbiomes from different populations, on one hand from wild populations that had within one species but different environments, and also from several species in the same environment, all from Dioun. And the question here is, if we want to test if the bacterial recruitment and maintenance of these microbiomes in the coralloid root is an environmental lottery, just anything goes in depending on where you are, or there's actual signatures of local adaptation of bacteria that are there. And so this is actually a very long story in itself that we're not quite finished analyzing yet, but I will anticipate or give you a sneak peek of what we've been finding so far, that not for all groups, but for some groups like cyanobacteria, we do have haplotype variation, as I mentioned before, of specific biosynthetic gene clusters that are associated with a particular function. For example, cytophore, which is transport of metals within the root, and tellurite-resistant haplotypes have variation according to soil type. So what you see here is a phylogeny that's constructed for cyanobacteria, and we mapped these biosynthetic gene clusters on each of the taxon that's in the phylogeny. So they're constructed independently, but what you can see here is that the distribution of genes basically corresponds to some of the clades that we recover with the phylogeny. So this is and this has to do with the traits of the soil that they're found in. We actually did measure all the pH and a lot of specific traits of the soil to see if there's something that we can link to the actual functions of each of these genes. And each of these clusters are a story in itself that I don't have too much time to go into, but essentially we're starting to find evidence that for at least some biosynthetic gene clusters, there is an local adaptation signal in some of the populations that we have. And one natural question is to see if they're under selection, or are they constrained, are they kept, what kind of selective signals they have. And we have started to look into this and find that it's actually quite a complex picture. Here we have several BGCs down here, different environments and different R samples compared to other cyanobacteria that are not symbiotic, or there are symbionts in different localities. So we're trying to make sense of this, but it does suggest that there's constraint to keep the function within certain groups according to soil type and place. So this is something we are still working on. So going back to this idea that everything has to be transferred, I did mention that in Xenia, it is conserved in the soil and in the roots. But what about the insects? So what we did is we looked at them. We knew from previous work by Shayla Salzman from the Naomi Pierce lab then at Harvard, that there's a taxonomic core conserved across all of the cycadivorous insects in the world. So basically they share a microbiome as we would have expected that they do in taxonomic composition. And so we're working with her, with Melissa Whittaker and with Naomi now to go in depth into these insects and see if they also conserve the function. So we have, we looked in 3 groups that are found in Mexican Dayun, in a butterfly, Eumeus childrenae, and Atala from Florida that we use for comparison, and 2 weevils that are the pollinators, strict pollinators from Dayun. And so what you see here is simply, we just compare different ways of looking into their microbiome, either by shotgun metagenome, 16S amplicons, or actual isolates from co-cultures, because we were worried that for a system as tiny as an insect gut, we were going to recover different things. So we actually, that was the case. And so we only picked 3 genera that we were sure that we would recover with all the different methods that we have here, which are the Stenotrophomonas, Serratia, and Pantoia, which are part of the core of these insects that were also recovered globally in the Salzman paper. And so looking at their functions, and this is again, the whole seminar in itself, but I will summarize that it seems that they're enriched for carotenoid-like metabolites and siderophores. And so what you see here is just a big scape analysis. Basically, these are networks. The nodes here are bacterial, different form, different shapes or different bacteria, one of these 3. And the fact that there's closed networks of these metabolites, predicted metabolites, means that they're not shared with any other bacteria from other systems from these 3 genera. So again, they're unique to the Cycad system, and they seem enriched for these types of metabolites. And we actually did HPLC. This is in collaboration with Paco Barona's group. We did HPLC to look for these metabolites and look to see their frequency and their abundance in all 3 genera. In different conditions, and we played with several other things. And we looked to see if they form networks, and from those networks, which ones are the most abundant and could be the ones that are key to have a function in this system. And this was done with mass spec. So we actually confirmed one natural question is, do the insects have BMAA in them? For example, among many other metabolites that I've mentioned, BMAA being the one that is perhaps the most important given the literature that I mentioned already at the beginning. And what we did is we did direct analysis real-time mass spectrometry to look for BMAA in the larvae and in the butterfly. And we found that BMAA is present indeed in the wings. So you can see here a graph where you can look at the amount of BMAA that we found in the wings compared to the gut. So this was good because that means that the insect really does recruit BMAA, tolerates BMAA, and so its gut microbiome has to be able to deal with this, which is, again, what we're finding indirectly. And we looked at this a little bit more carefully for Eumeus childrenae specifically. We chose this insect because it's the only one that actually chews leaves and ingests the leaves. The other ones just live inside the cones. And so we find that there's a core that we're looking into now and comparing with everything that I showed you before, especially with the roots and the soils bacteria that we think are transmitted in this whole cycad system. And we do find that as we expected, proteobacteria and cyanobacteria are present in this system. We still are not 100% sure what they're doing, but we have some insights, at least for some rhizobiales. We think that they might be essential for the color of the caterpillars, which has to do with carotenoid biosynthesis that I mentioned before briefly, specifically through a phytoin synthase that is unique to this system, as shown by this, again, phylogeny with map characters on it. And so this is something that we're following up now. There are many other stories that I could tell you, but my time is up actually 3 minutes ago. So I will leave you with these very brief take-home messages. Cycads do maintain specialized biological associations, and we think that these are key to their survival. They're not just in the cycad, they're transferred to their specialized multi-trophic interactions, and a combination of basically integrative biology, systems biology is the only, or perhaps the best way to understand this kind of complex system that has co-evolved in multiple ecological levels. So yes, this is one possible system that can allow us to understand the eco-evolutionary dynamic that maintains diversity over time. And with that, thank you very much. Here's a brief thank you slide with Prince Charles in a Cycad ceremony in Vanuatu. And this is the funding sources that have allowed me to do this work. Thanks so much.
Okay, Angélica, thank you very much for such a wonderful talk. I love it. I love particularly all the clear and beautiful pictures you show. So remember, everyone, you can post your question on the chat, both in English or Spanish. So I'm gonna check if there is any question here. Nothing for now.
No questions. Oh,
not yet.
Everyone's getting their coffee still.
We still have a few minutes, so we can wait.
Yeah, no problem.
Okay.
Well, we have like a minute.
Yeah. Here, Alexandra is congratulating you. Excelente charla. Muchas gracias.
Gracias, Alexandra. Well, I don't know if you have any questions, Juan. Digo, sí. Te iba a decir otra cosa. Do you have any questions?
Here, Patricia Lorena, also quiero felicitar a la ponente por la presentación, fue excelente. Excellent lecture.
Thank you. So what are the perspectives of the study? We're now looking at the metagenomes from the insect guts and also the roots and also the soil, basically from all the sources that I mentioned. We're doing more detailed comparative genomics and phylogenomics to confirm that these are actually transferred. So this is a very interesting idea. You know, I'm sure you've heard, well, we are what we eat, and so we as humans have microbiota or microbes that we eat from plants, from endophytes. And so that's the idea here, that insects also are eating the bacteria that are in the cycad, and when they die and when they go through their life cycle, they return it to the soil, and then the cycad gets to them again. So this is something we're working on. We also have work on fungi and viruses that I didn't mention. We think the fungi are highways to transport metabolites and bacteria rather than specific to the cycad. We think they're just more structural. And viruses, we think they're controlling the cyanobacteria, but we haven't really finished analyzing that yet. And so I do see another one that says, are these endosymbiotic species specific for cycads? Not the genera and not the order, not the big groups, not the genera, but the species we think they are. So we just sequenced, we used PacBio combined with Illumina to sequence one of the nostocs. We actually think it's not nostoc. I think it's we think it's desmonostoc, which is another genus. And the species is probably undescribed. So that's probably a species specific to cycads. There are other ones that have been described for cycads as well that are specific. But the genus and the order and the group is not specific to cycads. Nostocales are known for having multiple symbiotic interactions with many other plants and other organisms. So lichens and fungi and all sorts of things. Yeah, but their biosynthetic gene clusters are specific. So they're the same guys, or girls, but with different genetic functions.
Okay, I think we are running out of time, but we can address one more question. Here it says, quería preguntarles sobre la presencia del virus en Cycads. Yo trabajo con virus de plantas y he leído que en Cycads hay presencia de virus de DNA. Me gustaría preguntarle si tiene alguna información al respecto.
Sí, sí tenemos, pero te debo la lista porque no los recuerdo, pero tenemos una lista de virus que identificamos a través de transcriptomas de raíz. Y entonces hicimos la anotación y anotamos también cualquier otra cosa y salieron virus, pero con gusto te comparto la lista porque no recuerdo muy bien los nombres, pero sí tenemos virus de DNA y de RNA, los dos, que eso fue de RNA.
Okay. Another one, please. Mencionaste que las mariposas son los únicos insectos que se alimentan de las hojas. ¿Y los otros insectos que evaluaron interactúan con las plantas y cómo piensan que podrían incorporar las bacterias para que se mantenga el ciclo?
Sí. Es que sí se alimentan los coleópteros, pero del polen. Entonces, en el polen también hay, presumiblemente hay bacterias y definitivamente hay metabolitos. Y pues los tienen en los guts, o sea, ahí los tienen. También se comen el tejido de los conos, entonces seguramente ahí se están transfiriendo. Tienen todo su ciclo de vida dentro del cono masculino. Entonces, hay en algún momento donde se alimentan. Los adultos ya no, pero las larvas sí. Entonces, lo que mencioné, lo que pasa es que las mariposas son chewers y los otros tienen un estilo, pues son coleópteros curculiónidos con la proboscis. Entonces, hay una interacción distinta con la planta en términos de la cantidad de BMA o de otras cicasinas y tóxicos que podrían estar ingiriendo. Pero todos se las comen.
Okay, perfect. Well, we have one more.
Okay.
Bruno. Here in Brazil, it's way common domesticated cycads. Do you think the microbiome here will be also specific to our environment?
That's a really important question because Cycads is the genus from Japan and from China, from Asia, basically. So it's the commercial Cycads that you normally buy is Asian or of Asian origin. So one thing that we want to do is we want to look at Cycads outside of the range, but and see if they maintain the core. But compared to what has been published, the very few papers that have been published for cycad microbiomes, we do have the core is shared among all cycads. And what changes is the minor groups. So this is compared to another species of cycads in China. So I would expect that it has the same core, but it will have different satellite groups, and the function might be different though. I think the function will be different, and I mean that's interesting. We haven't looked.
Okay, perfect, Angélica.
Okay.
Thank you very much again, and thank you everyone for having came.
Okay, thanks so much.
Bye bye.
Ciao.